Hello, magandang araw sa inyo lahat. Ang didiscuss po natin ay tungkol sa cellular reproduction and genetics. So when you look at your parents, you can see the features that you share with them, such as the shape of the eyes, or the eye, the presence of dimples, or even the hand that you use when you write. The sharing of the features can be explained by heredity, where traits are passed on from parents to offspring. Okay, so when you look at your brothers and sisters, even if you share the same parents, each one of you can be considered unique based on the combination of traits each possesses. So that is what we call variation. Which demonstrates the difference among individuals. So when we talk about genetics, it is a study of heredity and variation. So it aims to understand how traits can be passed on to the next generation and how variation arises. Each living thing undergoes reproduction. So the nutrients taken by an individual will provide for energy. Okay, so provide for energy for metabolic processes, for growth and development, as well as reproduction. The cellular level of reproduction in the form of cell division. So provides the backdrop for the organismal level of reproduction. So this module will differentiate two types of cell division, namely mitosis and meiosis, as well as correlate heredity with the behavior of the chromosomes. Chromosomes in the cell during meiosis. So investigations are included that will help you understand the mechanism of heredity and expression of traits. So the question is, how are traits passed on to the next generation? Okay. Let us talk about the chromosomes. So living things contain what we call the genetic material that serves as a set of instructions that direct the activities and function of the cells. These genetic materials are also known as deoxyribonucleic acid, or what we call DNA. DNA are passed on from one generation to another generation to ensure that the continuity of life. In eukaryotic cell, eukaryotic cells, which are cells with organelles, the DNA are bound with proteins and are organized as beads on strings to form chromosomes. Okay, so I'll show you what the organization of the DNA as genetic material. Okay, so the number of chromosomes in a cell is characteristics or characteristic of species to which it belongs. For example, humans have 46 chromosomes, while rice have 12. So this is the list of selected organisms and their number of chromosomes. Let's talk about the cell cycle. The chromosomes of the cell change from as the cell transitions from one stage to another in a typical cell cycle. The cell cycle may be divided into two stages. The interphase, where the chromosomes are long and extended, and are also referred to as chromatids. In the cell division phase, where the chromosomes become condensed and thickened. So this is the cell cycle. Okay. The interphase refers to the period. Let me show you the okay. Hello. So the interphase refers to the period that follows one cell division and precedes another. So during this stage, the cell does not divide. It merely grows. The chromosome doubles or replicates itself because of the DNA molecule. DNA molecule contained in the chromosomes or chromosome produces an exact copy of itself. The interphase is divided into three substages. The stages from one formation of the new cell until it begins to replicate its DNA called the first gap. First gap period or G1. Okay, during which time the cell grows initially. The stage is characterized by protein and ribonucleic acid. Okay, which is what we call the RNA, which is synthesized based on the DNA, is then used to synthesize proteins. Okay, the middle stage is what we call the middle stage of the interphase is called synthesis stage or S stage. It's a period of DNA synthesis or replication. The chromosomes are duplicated in preparation for the next cell division. The second gap or the G2 falls between S period and M period, which is the cell division, mitosis and meiosis cell division. G2 represents the period of rapid growth of cell. Okay, for the preparation of cell division, of course, during the interphase. Okay, interphase pa rin. So during the interphase, the nucleus is clearly seen. Let us draw, let's say, a cell, and in the middle is nucleus. So interphase, the nucleus is clearly visible as a distinct membrane-bound organelle. So in stained cell, this membrane here can be clearly seen under the light microscope. Okay. One or more nucleoli inside it are visible inside, of course, the nucleus. On the other hand, the chromosomes cannot be clearly seen because they appear as an irregular mass that is grainy, in appearance because of the DNA they contain are stretched out thinly in the nucleus. This facilitates the replication of DNA during the S phase. Hello, check. Okay, alternating with the interphase is the cell division phase. In eukaryotic cell, there are two types of cell, namely mitosis and meiosis. Let us have first mitosis. So this type of cell division produces two identical cells with the same number of chromosomes. And this type of cell may be divided in, not maybe, but divided in 4 stages. So let us have the stage A, which is the prophase. So for the prophase, the nuclear membrane, now let me draw nucleus. Let's say this is a nucleus. Okay, the nuclear membrane and nucleoli may still be present. Okay, the chromosomes are thicker and shorter because of the repeated coiling. So at this stage, each chromosome is made up of 2 identical sister chromatids. Okay. As a consequence of replication of DNA during the S phase, so 2 chromatids produced from 1 chromosome are still attached at one point, called centromere. So let me draw a simple illustration of chromosome. Medyo balaktad, sorry. Okay, let's say this is the chromosome. Okay, the centromere here, okay, may divide we divide the chromosome into shorter arms called P arms, and then the long one is called Q arms. Okay, P stands for petite. Okay, in French. So, and the longer arms, which are the Q arms, the other one. Okay, so if the chromosomes are stained using GSMA, what the? Okay, let us divide or color them. With alternating two colors, black and white. Let's just assume that these color and uncolored are fully filled with colors. Okay, two colors, black and white only. Kasi ang hirap pulayan. Okay, we're talking about GSMA here. GSMA, alternating dark and light regions will appear. So, here. Okay, the black one is called heterochromatin. And then the white one is euchromatin. Okay, so let us go to the next one, which is the stage B, metaphase. So for the metaphase, the nuclear membrane, let's say this is a nucleus, nuclear membrane. Okay, so the nuclear membrane has disappeared. Okay. While the highly coiled chromosomes align in the at the metaphase plate, an imaginary plate equidistant between the cells, two poles. Spindle fibers are also formed. Each fiber binds to a protein called kinetochore at the centromere of each sister chromatids or chromatid of the chromosome. For stage C, which is the anaphase. So for anaphase, the paired centromere of each chromosome separate towards the opposite poles of the cells as they are pulled by the spindle fibers through their kinetochores. This liberates the sister chromatids. Each chromatid is now regarded as a full-fledged chromosome and is only made up of one sister chromatid. And for stage D, which is the telophase, the chromosomes are now at the opposite poles of the spindle. They start to uncoil and become indistinct under the light microscope. A new nuclear membrane forms around them, where the spindle fibers disappear. There is also cytokinesis on the division of the cytoplasm to form two separate daughter cells immediately after mitosis. Okay. And now we'll be talking about meiosis. What the? Excuse. The number of chromosomes normally remains the same within the species, so it does not double or triple for every generation. This suggests that a different kind of cell division must take place in an individual. So this kind of cell division is called meiosis, from the Greek word which means to make smaller. Okay, and it can be reduced. It reduces the chromosome number in half. So it takes place in plants and animals. Whenever gametes or sex cell are formed through the process of gametogenesis. Okay, meiosis is a special type of cell division where the cell undergoes two rounds of cell dividing, two rounds of division, okay, to produce four daughter cells. Okay, so each with half with the chromosome number as the original parent cell with a unique set of genetic materials as a result of exchange of chromosome segments during the process of crossing over. Okay, the first round of meiotic division is also known as Meiosis 1. Consists of 4 stages. Namely, prophase 1. Prophase, next is metaphase. Next is anaphase. And lastly, the telophase. And they are under meiosis 1. Okay, prophase 1 of meiosis 1, and unlike its counterpart in mitosis, is more elaborate and should be understood well in order to grasp the mechanism of heredity. Let us have prophase 1. Meiosis starts with a stage and includes the following sub-stages. Okay, the first sub-stages of prophase 1 is the leptotin. Leptotin, each chromosome is made up of 2 long threads, okay, of sister chromatids as a result of replication during TS phase of cell cycle. Next is zygote. Chromosome is made up of 2 long threads. Chromosomes begin to pair. Okay, pair of chromosomes are called homologous. Okay, and this pairing of chromosomes, this process, okay, is exact. Next is pakitin. The chromosome contract due to the repeated coiling. Okay, the chromosomes contract due to the repeated coiling. Crossing over takes place during the stage where the segment of sister chromatids of one chromosome is exchanged with the same segment of the sister chromatid of the homologous chromosome through the formation of the cross-linkage of the segment called chiasma. So after crossing over, the sister chromatid of each chromosome may no longer be identical with each other based on their genetic material they contain. Let me draw the crossing over of homologous chromosome during meiosis 1. Okay, so this illustration, even though it's not as good as this expresses the crossing over of homologous chromosome during meiosis 1, wherein we have homologous chromosomes here, and then this illustrates or explains the crossing over. Okay, and then it results to combination. Okay, let us move to the next. The next stage is diakinesis. Sorry, not diakinesis, diplotin. In diplotin, the chromosomes begin to uncoil. Okay, next is diakinesis. Diakinesis, paired chromosomes dispersed. In nucleus. Okay, that's it for what is the first stage? Prophase 1 of meiosis cell division. Let's go now to the stage B, which is metaphase 1. Okay, the paired chromosome arrange. So arrange themselves along the equatorial plate. Next is stage C, which is the anaphase 1. Pagandang sulat ko. Spindle fibers form and attach to the centromeres of the chromosomes. Okay, as you remember, we already discussed what centromere is. That is the center of the chromosome. Okay, wherein it can divide the chromosome into two. Okay, so spindle fibers attach themselves to it. So the homologous chromosomes separate from each other completely and start their movement toward the poles of the cells as they pulled by the spindle fibers. As the centromere of each chromosome does not divide, the sister chromatids remain together. Okay, next. Stage D, which is the telophase 1. This is a stage where the chromosomes reach their respective poles. Okay. Cytokinesis follows and the two daughter cells are formed. Let me write some info. Okay, and then cytokinesis follows. Two daughter cells are formed. Okay, each cell now has only half of the chromosome number because only one chromosome from each pair goes to the daughter cell. This is called haploid. Haploid condition, in contrast to the diploid condition at the beginning of meiosis 1, where each chromosome pair is intact. Telophase 1 is followed by interphase 2. Telophase 1 is followed by interphase 2. Okay. Note that each of the chromosomes still has two sister chromatids. It is therefore necessary for the cells to undergo another round of cell division. So the second meiotic division is also known as meiosis 2. Is mitotic in nature and consists of the following stages. We have prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, so these stages are identical with the mitotic stages. The results are 4 cells. So the first process of division, okay, meiotic division, I'm sorry, will undergo next division, which has the same division. Okay, yung nauna parehas lang din. Uulitin lang ulit. Okay? So the results are 4 cells to form each daughter cells from meiosis 1 with 1/2 of the diploid chromosome number with only one sister chromatid for each chromosome. Now let me show you the picture of the division of or stages of meiosis in certain plant. Okay, I guess we're done discussing cell division, 2 types of cell division. I hope you like it and you do enjoy listening. You may like, share, and consider subscribing for more updates. Thank you.